The first step in culturing microalgae is to set up the phytoplankton culture equipment. After setting the equipment, make the phytoplankton culture media. Culture media is the saltwater solution where the plankton will grow in. To make culture media, add salt into the fresh water and stir it before leaving it overnight. The culture will start from a starter culture in 500 ml bottle, and the culture will be transferred from container to bag culture. To prevent bacterial contamination, the water is sterile by running the water through UV sterilizer. For grow-out process, 1/3 of the culture is poured into 2 new bottles and harvest 2/3 of each bottle. The remaining 1/3 of the culture in the original bottle is topped off with new media. Harvested algae can be filled in empty bottles to store it in refrigerator for further use. 1 drop of mixed algal suspension is put on each new blower chamber and cover it with the cover slip. First, start counting at the top left square and count cells that only lie within or touch the right and top boundary lines. Do not count the bottom and left line to avoid counting the same line. The cells in squares that spread randomly in the counting area is then counted. Rotifers, a microscopic aquatic animals, are a great food source for newly hatched fish larvae and for some filter feeding invertebrates. For the first step of culturing rotifer, prepare a bottle or container. After adding the rotifer starter in the bottle, mix the solution with seawater before installing the acetone into the culture for their oxygen supply. Make sure that the aeration is always on and wash the culture over each of the next few days. To harvest the rotifers, pour out about 3/4 of the tea-colored culture through a rotifer sieve into a water change bucket. Microworms are tiny nematodes with the perfect size of food for newly hatched fry. They can also be fed to ornamental fish that are too small. Few things needed for microworm culture are container with lids, spoon, oatmeal, microworm culture starter, and active dry yeast. First, poke small holes on the container lids to let air in. Next, add water into the oatmeal and stir thoroughly and make the best meat. Leave oatmeal to cool overnight in room temperature before adding microworm culture into the oatmeal. Last but not least, add active dry yeast as food source for microworms. Mix the culture once a week using a spoon to keep it fresh. For harvesting, wipe the side of container where the worms are crawling with a rubber spatula or a Q-tips and feed it to the fry.